Thank you guys for coming. So any burning questions before we get started? I think someone might have their mic on. If you don't mind muting yourself just for background noise. So the first thing I want to say about the tissues, I know I had shown you this before, but I'll just repeat this because it is pretty important. So in your guys's study resources module, you have access to a few things that are really helpful for both exams, and then we're talking about the tissues. So you have this lecture exam number 1, practice problems, and then tissues checklist for lab exam number 1. If you guys click on that, it basically pulls up this document, and this document is like a study guide of what you should know for the tissues portion of the lab exam. Okay, so I guess we should rename it Tissues Study Guide for Lab Exam 1. Does that make sense? So right here, Tissues Checklist for Lab Exam 1 is really like a study guide. If you read through here, it tells you what you need to know for each tissue found in the packet. Okay. Then right below it, lab exam number 1, practice exam PowerPoint. That's what I'm going to be using. We're going to be going through that right now with additional pictures to give examples. And then remember that if you guys go to your actual packet, this lab exam number 1 tissues packet, if you're in vendors class, it will be where you would submit your packet. If you go there, it pulls up this Google Drive link that includes additional pictures. Okay, so I'm going to use 3 different basically resources. Does this make sense? So these are all like additional resources kind of. They're all built into your module. So check out your study resources module. All of you guys should have access to the study resources module, whether you're in vendors class or Knutsen's class. And then your module will look a little bit different, but both of you will have access to this same Google Drive link. Okay. I am not able to give permission to record the Zoom meeting because I don't own the Zoom meeting. So you can try to ask the person who let you into this breakout room, Dan, to see if he can. I'm not sure. I've asked for permission to record before, and I was never able to, but maybe that was a technical error on my part, so it wouldn't hurt to ask. I think you might have to leave the breakout room and then come back in. So yeah, I don't own the meeting, so I'm not able to give permission. I don't know that they will though, because anyway, you might as well ask, but it's already being recorded, so I'm not sure if you can double record. Okay, so I kind of had mentioned, right, to you guys for you to start filling out the tissues lab packet over this past weekend. So hopefully you guys made some progress on that, kind of have started to fill it out. Remember that filling it out is as simple as answering the questions, each question, and then maybe making a drawing. Okay, so not too complicated to fill that out. Now, when we look at comparing and contrasting each of the tissues, there's kind of some tips and tricks to tell them apart, tell each of the tissues apart. How can I turn on subtitles? I'm not sure how to turn on subtitles. That's a great question. If anyone has an answer for that. I don't know. I didn't even know you could do that. I'm not sure. So basically we'll start with our simple squamous epithelium. Okay, so with the simple squamous epithelium, this is found in the lungs. So if you go to our checklist, and I'll just show you this so you guys can kind of see, but if we go to the checklist right here, what do we need to know for for the lab exam? What are you guys responsible for? You need to know the name of the tissue. You need to know that the air sacs are also called alveoli. And then you should know how to tell the difference between frog skin and lung tissue, and then the location. So where is simple squamous epithelium found? In the lung, right? And then what are our alveoli or air sacs? These open circles here would be the alveoli or the air sacs. Alveoli, okay? So air sac is the other name for it. Good? This is just an example here of frog skin. Any questions there? I feel like that one's probably something everybody's taken a look at, right? So how would it look on your lab exam? Do you guys see this? I'll show you this in this picture because I feel like it's probably a little more straightforward. Do you see this black pointer? That's what you guys would see on your lab exam. So if we were asking you to identify a part of the tissue, we would have the black pointer pointed to it. Like, for example, here, the black pointer is pointing at a goblet cell. And then the question would say something like, what is the structure at the end of the pointer? Does that make sense? So that's how you can expect to need to identify these additional kind of special terms. I call them special terms. So if you look on here, what is the special term you need to know for simple squamous? It would be alveoli or air sacs. So what that means is if you're on the lab exam, we might have a pointer directed at these open white areas, right? And it would say, what is the structure at the end of the pointer? And then you would need to identify alveoli. Does that make sense? So we could ask you, identify this tissue, and then you would need to be able to, you should prepare for it to be a fill-in exam because there might be fill-in questions. So identify this tissue, you would write simple squamous epithelium. You need to know the full name, not just simple squamous or not just squamous, it needs to be simple squamous epithelium. And then where is this tissue located, right? And then our answer here is lungs. And then what is the structure at the end of the pointer? Alveoli. So those are kind of the three questions you can get on any tissue. So we'll keep moving through our tissues. So for simple cuboidal epithelium, this one kind of looks like someone took a hose or a little tube and cut it into donuts. You can see that they all kind of look like these little donut shapes over and over. So this is simple cuboidal epithelium. It's located in the kidney. So like that's why I put it's fine if you just know kidney as the location. And then characteristic is kind of a way for you to remember what it looks like. So this looks like these open white circles enclosed by dots. So basically donuts, right? Donuts of little dots. And the way the slide is cut sometimes make the circles look like they are these weird long shapes, but they're still a circle. So it's still supposed to be just circles. It's just that sometimes it's cut at a weird angle. So these don't really accurately represent what the kidney tubules look like. The kidney tubules look like this. So what would you be identifying for this tissue? You would be identifying the name, simple cuboidal epithelium, and then the location, which is kidney. Does that make sense? Name and location. That's what you would be identifying for this tissue on the exam. So these two were kind of easy, right? We kind of feel like we get the idea of how this looks. Sometimes when students are first learning, they typically all get the question, how do you tell the difference between this simple cuboidal and this simple squamous? They both have kind of these openings, these circles. So let me pull up another example or two and then hopefully it'll clear it up. So this is lung tissue looked at from a lot further away. So in this example, lung tissue, this is simple squamous epithelium, right? Oh, all right. I will see you guys out there in the main session, okay? And we'll keep going.
Recording in progress. Hang on just a second. Got people jumping in right now.
I'm here, right?
Excuse me.
Yes, you're here.
For those of you who just came in, I just need to know your first and last name and Delta ID. You can just type it in the chat. Rosalinda, can you type your Delta ID in the chat? Anyway, so for those of you who have just come in recently, just type your name and ID in the chat. That way I can follow up on it after the fact. I'm going to pass over to Brooke, and then I'm going to take off.
Perfect. And as usual, if anybody needs to leave early, just make sure you put your say something in the chat about leaving, right, Dan?
Yeah.
Okay. That way with it, he can record the time that you're leaving, basically.
Yeah.
All right, thank you.
Mm-hmm.
Okay, so the tissue we're looking at right now is basically lung tissue seen from really far away. So when you look at it, it has this appearance like open circles, right? So when we zoom in on lung tissue really close, it looks like this. It's like that little circle, but really close up. So this is far away, and then this would be really close. So the idea here is that you're trying to figure out, why does this lung simple squamous epithelium, why does this look different than simple cuboidal epithelium, okay? And if you look at it from far away, it makes sense, right? It's kind of crazy all over the place, little lines everywhere, and it's not really consistent and perfect circles. Versus if you look at our simple cuboidal epithelium found in the kidney, right? You see these perfect circles. You see what I'm saying? Perfect circles with these little cells around it. Versus our simple squamous, again, is our not perfect circles. They're kind of like irregular in shape. And they really do look like they have more than one layer of cells, but they are simple. So technically it's just one layer, okay? So for this tissue, it's simple squamous epithelium. It is located in the lung, and the open circles are called alveoli or air sacs. The blue writing would line up with simple cuboidal. This tissue that we see here is simple cuboidal epithelium, and it is located in the kidney. Okay, so hopefully that helps identify the difference between those two tissues. One more example of our simple cuboidal epithelium is right here. So this is showing you that in this picture, they are all oblong, but as you can see, even though they don't look like donuts here, they are oblong in shape, and they do have that kind of those cells going around that open circle. But it would be very unlikely you would see this on your lab exam. You're going to get a picture that looks similar to this in the sense that you will see these circular shapes. Just know that the dye we use on the microscope slide may not be this color. So we may use this pink dye, but the appearance should be very similar to this with these circles lined by these cells, okay? So moving on to simple columnar epithelium. So for simple columnar epithelium, we're going to see what I call columns of cells. So I think that this looks like a stack of like cells standing next to each other going around this villus. So I'll kind of try to break it down what I'm talking about. So do you guys see how we kind of track this line of cells, right? In between my red lines, do you see how it's like these closely packed uniform cells that go all the way around this like big giant finger structure? Yes? Is that kind of, hopefully you're seeing that. It's kind of takes a little bit of imagination at first and you feel like you're really pushing your imagination, but it is there. So I'll show you another example. And then we'll kind of get into the different special structures with it. So when we zoom in on it, because this is our same slide, but I'll zoom in. You guys can see how we have all of these cells in a stack and they just kind of follow along that villus line. So the finger structure, this big kind of object, this whole giant thing that's sticking out, there's another one down here. Do you see them? They're like these big claws or fingers. These are called villus. So the name for these is villus. Villus, multiple, we call them villi. So villus, villi. And the function of the villus or the villi that you know for the test is that they increase surface area. So the villus or the villi increase surface area, okay? What tissue is this? This is simple columnar epithelium. Where is it located? This is located in the intestine. So this lines your intestinal tract. What is in a person's intestines? Isn't it the food you eat, right? When you eat something, it goes into your intestines. Yes? I know this is just the beginning of the class, but we all kind of know that at this point, I think. So what are our special terms we're going to identify? So we want to identify villus, goblet cell, and I'm going to do these right now. Villus, goblet cell, microvilli, and we need to know their functions, okay? So this is really important for this tissue especially. We need you guys to know these functions. Villus, goblet cell, microvilli, and then you should know the function of the simple columnar, the lacteal, and then the capillary bed. So I'll show those to you here. So we just labeled the villus, right? It's like these big finger-like structures and their function is to increase surface area. And then lining the surface of the villi, I'll use yellow. So do you guys see this little pink line here? It's like this pink blurry line and it goes all along the surface of the villi all the way around. Yes? Pink kind of blurry line right here. That represents the microvilli. Okay, so the microvilli are little tiny, they are like hairs, but they're more like bristles of a toothbrush, maybe. You could think of them like bristles on a toothbrush is a good way to like think about it. That's not going to be on your test. It's just a way to remember what they're kind of the texture. They're like stiff and they slow food down for absorption. So when food, let's say you eat, you eat your breakfast sandwich. Now the food is basically going to pass, let's try to make it a standout color. So this is your breakfast sandwich that you ate. It's now in your intestine, right? Because the location of this tissue is the intestine. So what that food is going to do is it's going to now make its journey along these villi. And as it's kind of passing through, to slow that food down to allow it to actually, the particles, we want the particles of the food to get into your blood, okay? So waiting right here is the capillary bed. So the capillary bed is at the center of the villus, and that will actually absorb the food. So the capillary bed absorbs proteins, carbs, and nucleic acids. So those are part of the components of the food that you eat. The other thing that's sitting inside of this center divide, and yes, this is all stuff we could test you on for the lab exam, just to be clear. The other thing that's in here is the lacteal. So there's two structures that are sitting in the middle. One is, again, the capillary bed, and then one is called the lacteal. So the lacteal absorbs fatty acids. So your breakfast sandwich had some bread on it. Where is the bread absorbed? In the capillary bed. Does that make sense? Carbohydrates. Your sandwich had some meat. That would be protein. It's absorbed by the capillary bed. And then maybe it had fatty acids or fat, and that would be absorbed by the lacteal. Okay, so the microvilli, we got that function. Villus, we get that's the overall thing. Lacteal is inside of the villus, and then the capillary bed is as well. And then we have our goblet cells. So each of these little round white circles that you see, each of those represents a goblet cell. And the function of the goblet cell is to secrete mucin/mucus to keep the intestine moist. So it's basically keeping the lining of the intestine moist, allowing the food to be able to glide or slide along that area. Okay, so where are the simple columnar cells? Just to make sure we're really clear on that. This row right here that goes all the way around, right? This row of cells, those are the simple columnar cells. So the simple columnar cells are basically this outer row of the villus. Okay. Any questions on this tissue? This is kind of a involved tissue, and we're about to contrast it to the one that students confuse this with, and I'll show you how to tell them apart. Okay.
I have a question real quick. How do you tell the difference between the lacteal and the capillary bed? Like,
between the lacteal and the capillary bed. So on our microscopes, you can't. You might be asked, what absorbs proteins in this area? What absorbs fatty acids for this tissue? So you might be asked like that. Does that make sense? Kind of like a definition.
oh, okay, so it's not gonna like point to the middle and be like, oh, what is this? And ask for the lacteal or the capillary bed.
No, you don't need to worry about that.
Okay.
But you do need to identify those and like tell them apart on a model. So one of our models is like an intestine model, and I'll try to pull that up either today or Thursday. But that one you do need to actually say, okay, that's the lacteal, that's the capillary bed, but not on the microscope.
Okay, thank you.
Yeah, so someone asked the microvilli versus the villi. The villi is like this entire, I don't know, it looks like a tower, maybe like this big, massive thing. I don't know, I'm trying to find a color that I haven't used. So I'll just draw a circle around it. This whole entire thing that I drew a circle around is the villi. Does that make sense? It's really big. The microvilli is this pink line that goes along the outer surface of the villi. So this would be micro. Think about it. It sounds like it's small, right? Micro, like a small thing, versus villus is a big thing. Villus is the whole thing. Micro is on the surface. All right. I don't know if I missed any questions in the chat. If I don't see your question, just try to ask it out loud if I pause for questions, but I'm trying to be looking at the chat throughout it, but sometimes I get kind of behind on checking the chat for questions. So don't be afraid to verbally ask. I think everybody in the chat looks like, yes, you need to know certain functions. Someone asked me about functions. Refer to this special terms checklist to get whether or not you need to know a function. So for example, like here, I make a very clear point of showing you guys know their functions. Does that make sense? Versus like here and its function. Some of them don't always, it's like basic function versus like detailed functions. There's no functions here. So like you don't need to know the function of fusiform. You do need to know the function of intercalated disc. Does that make sense? So refer to this if you're wanting detail on which functions you need to know. And I'll try to do my best to cover as much as possible here in a second. So this is another example of simple columnar epithelium. So this was simple columnar epithelium. This is another example of simple columnar epithelium. Hopefully you guys are like feeling a little bit like, whoa, those look really different from each other, right? How would I tell those apart on an exam? Well, let's look at a few things that we can see right off the bat. Do you guys see the goblet cells? Everybody's seeing the goblet cells, those white circles? That is a big, big giveaway that this is simple columnar epithelium. We're about to look at one other tissue that has goblet cells, but the goblet cells should be a big key for you. The other thing you can see here is basically it kind of looks like the villus, right? It looks like we took a microscope and we zoomed in on like just this part, and now we're looking at it really closely. Does that make sense? So sometimes you have to kind of get your brain outside of the box of like looking at it from one view, but I will try to encourage you that we're not trying to trick you on the exam. It's just, we're trying to test your knowledge, but we're not going to like, I don't know, zoom in on like the time. This is honestly, this is too zoomed in. I wouldn't put this on a lab exam for your question because I do feel like this is too zoomed in, but it might ask you, it would be a fair question if I was wanting you to identify goblet cell, right? Because like a goblet cell is a goblet cell. Does that make sense? It's not, there's nothing else that looks like these weird open white circles in a tissue. All right, so let's contrast this with the one it usually gets confused with, which is ciliated pseudostratified columnar epithelium. So this picture is very blurry, but I actually chose it on purpose. I know it seems like kind of weird that I would choose a blurry picture, but I like this picture because it shows you guys how there's only one layer of cells. So I'll show you another example of that. So in ciliated pseudostratified columnar epithelium, very long mouthful of word, but there's only one layer of cells. So I'll try to find you yet another example, see if we can drive the point home. So do you guys keep this in your head? Like take a little mental picture and try to hold it in your head. Do you remember we have a layer wrapping around a villus, right? So let's go to our other one. So that's kind of like we can almost see two layers of cells, right? Ciliated pseudostratified columnar epithelium, you only ever see one layer. And that has to do with the location. So ciliated pseudostratified columnar epithelium, the location is lining the respiratory tract. So this is in your throat right now. And it basically has these the special term or special structure is cilia, and cilia move mucus upward. So on the surface of our ciliated pseudostratified columnar epithelium, do you guys see these little hair-like structures right here? Kind of look like somebody took, I don't know, like peach fuzz. That's what it reminds me of. So cilia are hair-like structures that move mucus upward. This is a goblet cell right here. So it's the same goblet cell we would see in our simple columnar. This goblet cell also secretes mucus. Now, what do you guys know about mucus and respiratory tract, right? When you guys get sick with a cold, you like cough up mucus, or maybe you go out and you get dust in your lungs and you cough up like phlegm with dirt in it. That's because the cilia are pushing up that mucus that has the dirt particles or maybe bacteria, white blood cells, and it's pushing it up so you can cough it up. Does that make sense? So the function of cilia is to move mucus, right? So the big ones that people get confused on the exam is cilia versus microvilli. Microvilli slow food down for absorption. And the way you can tell the difference between cilia and microvilli are not visible as hairs on the microscope. So do you guys see how this looks like hair? Yes, cilia looks like hair. I'm gonna go pull up another one here. So this is another example of our ciliated pseudostratified columnar epithelium. And do you see the little hairs here? How we can just make out kind of like these little individual hairs, like dots. But when I go back to the simple columnar epithelium, remember our microvilli, all it looks like is a pink blurry line. There are no hairs, so we wouldn't count this little weird thing as a hair. There are not hairs. So how do you tell the difference between ciliated pseudostratified columnar epithelium and simple columnar epithelium? The number one way is to look for the hairs. If you see hairs, it would be, if you see hairs, it's ciliated pseudostratified columnar epithelium. If you don't see hairs, it's likely to be simple columnar, okay? The other way you can kind of tell them apart is, so I guess I can write this over here. How to tell apart. If you see hairs, like individual little hairs, that would be ciliated. And then if you see 2 columns of cells, that would be simple columnar. So do you guys see how we have column number 1 here, column number 1 up here, and then column number 2 down here? Yes, those are 2. Our other example that I showed you over here was this guy, right? 2 columns of cells. Do you guys see 1 and 2, right? But ciliated pseudostratified only has 1 column of cells. So right here, we have 1 column of cells. On our next picture here, we also have 1 column of cells. Is that making sense? So again, 2 columns of cells for simple columnar, and the microvilli is not hairs. Like you can't see hairs for microvilli. And then for ciliated pseudostratified, you can see the hairs, and we only have 1 layer of cells. Good? I know there's a lot, and I'm trying to move fairly quickly. I'm not trying to, I'm hoping that I'm not breezing past it. I'm trying to cover it, but at the same time, I'm trying to get through a lot. There's 24 tissues. So I'm doing my best to go methodically. So any questions on that? So that was ciliated versus simple columnar. Those are often confused by students. Okay, and I have some of that stuff that I'd written down, I have here, okay? Function of cilia, function of goblet cells. All right, so let's do transitional epithelium. So transitional epithelium is another example of an epithelial tissue. It has a variety of tissues kind of seen on one slide. So sometimes this does get a little confusing for students. The biggest area that you guys are looking at, so where our actual transitional epithelium is right here. So what is outlined, what I just outlined, that's the transitional epithelium. So transitional epithelium, the main place we think of is our bladder. So this means that urine fills this cavity right here. So if you picture it, if urine was filling this up to the point where someone really needed to go pee, right? The urine is starting to fill these pockets, and basically this is expanding bigger and bigger and stretching out. So the reason it's called transitional epithelium is it transitions from basically all of these cells kind of like hanging out, relaxed, and then they will transition to being one tight singular layer of cells when the bladder is full. Same thing with the uterus, right? When there's no baby, it's going to be very relaxed. Lots of cells, looks like a lot of cells all sitting together. And then when a baby actually is growing, we're going to see a little line of cells around the outside. So transitional, because it transitions from a lot of packed cells to a single line. The function of that is really important to being able to remember what it looks like. Does that make sense? Because we're kind of remembering that they look like a lot of weird little cells packed on top of each other. You might be looking out here and you're kind of confused, like what's all this other tissue doing here? This is smooth muscle. So smooth muscle helps you to pee. So if you go pee, right, you force your bladder to empty out. That muscle is inside forcing your bladder to empty out. So that is smooth muscle that's doing that, forcing that bladder to empty, okay? So our characteristic here, how we tell that this is transitional, we're gonna look for that circle on the outside of smooth muscle, and then we see these wavy packed together cells on the inside. And this is what I was kind of explaining. It changes shape depending on whether it's stretched or relaxed. And we see a relaxed bladder here, but it will actually expand to change the shape of the cells. So right now they're stacked, but then they flatten out as the bladder fills up. So transitional, I would say, is not typically confused by students with other tissues unless you just didn't study this one, which does happen. Maybe, yeah, I don't really know what you could confuse it for. It's basically very distinctive. Again, if you look at it under a microscope and you actually take your time looking at it, you shouldn't be really confusing it with anything else, okay? But hopefully you're realizing if you do confuse it with something else, spend that time studying and figure out, okay, well, why does it look like this tissue? Why does it look like it? And there is an answer. There's always an answer. And I'll share you my story in a second. So here we have stratified squamous epithelium. Someone in a previous SI had asked me about the difference between pigmented and non-pigmented. So pigmented, where you're going to look is right here. Do you guys see this? All of these brown, this is a bad color. Do you guys see all these brown cells right here at the bottom? So this layer of the skin is called the stratum basala. So right here, stratum basala. And it is how we determine pigmented versus non-pigmented. So we look for the cells in the stratum basala, and that tells us if it's pigmented or non-pigmented. If they are brown, it's pigmented. And then if they're not brown, that would be non-pigmented. So for an example of non-pigmented, I think I have one in here. This would be non-pigmented right here. So this is a non-pigmented example. I know that it has a few brown cells, but when you see our truly pigmented example, it's very clear that it's pigmented. Do you guys see how clearly pigmented this one would be? So one of the things you guys need to be able to do for your packet and then also for the lab exam is label the layers. So we just said that this layer is called what? Stratum basala, right? So that's the way you guys can remember the layers is boys, and then we can go boys say, girls like chocolate. Okay, so this chocolate is the top layer, it's the stratum corneum, and you do need to know how to identify these on the lab exam. So on the lab exam, you do need to know how to identify each of these layers. So I'll go through and I'll label them. Stratum granulosum for girls, stratum spinosum for say, and then stratum basala. So boys starts it, and that's the bottom layer. So boys basala is the bottom layer. So this, we're going to write a B. Let's see, what can I get my? So this is B for boys. I don't love that. That color is really hard to see. Let's just do black. Okay. Next one would be stratum spinosum, right? Say, boys say. So stratum spinosum is right here. Okay, this is S, stratum spinosum. Good, stratum spinosum. Granulosum is the line right here. So this line at the top, kind of basically where we turn from the pink side to kind of the more light side. This is G, granulosum. Boys say, girls. So next we have like and then chocolate. This is a thin skin sample. So in thin skin, Lucidum is not present. And I will show you guys an example of thick skin, but for now, let's just finish labeling this. So all of these little folds or little lines that look like hairs, those are corneum, okay? So corneum are all of these little lines or hairs right here would be corneum. So basala, bottom layer where all of those pigmented cells are. Spinosum would be kind of the pink. Granulosum is this line at the top. And then corneum are our little hair-like cell, hair-like layer. So I'm going to label the layers again, but this time on a thick skin slide. Okay, so let's go to a thick skin. So right here is our thick skin example. So if we look at that, we can see our layers again, but it's really far away in comparison, but we can still see all the same structures. So do you guys see how we have this curviness? All of those curves, that would be our bottom basala layer. Everything in between, this like light pink, is spinosum. Boys say, right? And I know my thing is white, but boys say, and then G or granulosum is this purple line. Now, this is a thick skin example. Lucidum is this purple, like light purple layer, lucidum. Corneum is all of this thick layer. So how do you remember the mnemonic? Again, it's boys say, girls like chocolate. Remember, like or lucidum is only in thick skin. They all start with stratum. Boys is stratum basala. Say is stratum spinosum. Girls is stratum granulosum. Like is stratum lucidum. And then C, chocolate is stratum corneum. You don't need to know functions. You just would need to know how to identify them. Okay, so how do you identify which one? If I have a arrow pointing, right? Because remember, you're going to have a pointer within the microscope on your lab exam. So if I have a pointer pointing at this light purple area, what is the structure at the end of the pointer? So what is the structure at the end of the pointer? And you need to know that this is our stratum lucidum. Does that make sense to everybody? Kind of how to expect the lab exam to be? Labeling what's at the end of the pointer. Also identify this tissue. So what tissue do we see here? Stratified squamous epithelium, right? Where is this tissue found? The skin, right? This tissue makes up the human skin.
So that
You cut out. Sorry, were you asking something? I think Vanessa. All right. Any questions at all? All right. So again, with typically students get confused on this tissue if they don't study this tissue. So this isn't really one that looks a whole lot like other tissues. It's more like if you don't spend your time studying it, you're not going to know what it is.
I just have a quick question.
Yeah.
What is the white part in that one that kind of looks like birds?
Oh, that's a great question. I'm pretty sure it's just when if you picture like cutting off part of the skin, I think it kind of tore. It looks like it tore a little bit when they put it on the slide.
Oh, okay.
Sometimes things can look like something and it's really just when they cut it and put it on the slide, it did it. It's possible it's something, but it's nothing you need to know. So, good question.
Okay, thank you.
Yeah. So in this one, this picture, we see stratified squamous epithelium again, but this kind of shows you guys a few different tissue types. So remember our stratified cuboidal, right? So right here is our stratified squamous epithelium. do you guys see the little wavy line? That would be our boys. I'm trying to write it really small. This would be say, and then granulosum is at the line, and then corneum would be somewhere up here. So you see, we're picturing skin, right? Look at your skin and think about all the things that are sitting underneath your skin. You have hair follicles, you have sweat glands, you have muscle deeper than that, tendons. So it keeps going very deep. So these here, the glands that are found surrounding the hair follicle are our sebaceous glands. So the sebaceous oil gland surrounds the hair follicle, and it basically puts out oil to keep it moist. So the function is to keep the hair follicle moist. What type of tissue is it? So if on your exam we were pointing to it, what type of tissue is it? It is stratified cuboidal epithelium, right? So if on the exam we have a pointer pointing to one of these little round white structures, you would need to know stratified cuboidal epithelium is the tissue. They are a sebaceous oil gland keeping the hair follicle moist. Okay. And like I have here, not in the picture, are sudoriferous sweat glands. We don't really have a picture for you guys of the sudoriferous sweat glands, so you're not going to get tested on the microscope on sudoriferous sweat glands. So sudoriferous, not tested on the microscope. We do have a model, so just be ready to answer about that on the model, but it's not going to be tested on the microscope. Any questions?
I have a question.
Yeah.
For the sebaceous oil gland, like how can you tell that it's stratified cuboidal? Because to me, I can only see like the white parts, but not the actual like cube-like shapes.
That is a great question. Really, with a lot of these, sometimes you can't even tell. Like sometimes it's supposed to be named whatever it's named, and you can't tell that it is because of different reasons, either the way that it was cut or the way that our microscopes are maybe not high quality enough to see it. In this particular example that we're looking at right now, it's because the microscope isn't zoomed in enough on just the stratified cuboidal patch. So like if we looked really, really closely and we had a high powered microscope, you would be able to see that they look like little cuboidal cells, but you're just supposed to know based off of the location if they're found around a hair follicle, and you guys can see here that this is a hair follicle, then you know it's a sebaceous gland. Does that make sense? Do you guys see this big structure here? That is your hair follicle. So if it's a gland found around the hair follicle, there's no other option except for it to be a sebaceous gland. And if it's a sebaceous gland, it's stratified cuboidal. Does that make sense?
Yeah, that makes sense. Thank you.
Okay. So we're going to keep going here. So the next one, our next little tissue is our Pacinian corpuscle. You will, I don't know which of these you have on the test, but we always do at least one, if not both of these on your lab exam. So the Pacinian corpuscle is found deep inside of the skin. So I put that here, deep in the integument or skin. And its function is to detect pressure. Okay, so that's the function you would know for the lab exam. I'm going to go here because we have a better example of the Pacinian corpuscle where I can actually zoom for you guys to see it. Yeah. All right, so let's zoom in here. So do you guys see how it almost looks like somebody took an onion and cut it? Do you see all of these like rows and lines of cells kind of looping around? So that is our Pacinian corpuscle. Again, Pacinian corpuscle. Function, detect pressure. Location, deep in the skin. okay? And it kind of has this appearance looking something like this. Yeah? Good. So again, no real tissue you can get it confused with. Students who get this wrong are usually students who didn't study it. Again, what do you need to know? Pacinian corpuscle, you do need to know the function. You're very likely to be asked both. If you're asked them, they'd be asked together, okay? Pacinian corpuscle and then the function. If you're wondering where to see the slides that I'm doing right now, the slides I'm going over right now are found in the study resources module under practice exam PowerPoint. That's where these slides are. All right, so for Meissner's corpuscle, so you guys see that it kind of looks like a bean. So the way that I remember this is a bean-like shape. So as it says here, you need to know the function that it detects light touch and that it's found in the papillary layer of the dermis. So name is this Meissner's corpuscle. Function, it detects light touch, and then location in the papillary layer of the dermis. So where is the papillary layer of the dermis? So let's go back in time a little bit and we'll show you on this picture. I like this picture a little better. So this right here, you can actually see a Meissner's corpuscle in this picture. This is a Meissner's corpuscle visible here. You would not need to identify it on the lab exam that far away. So don't panic. You don't identify them from this much of a distance. But the papillary layer of the dermis, you do need to know. So what is the papillary layer of the dermis? So this portion here, well, technically all through this, what is that called? That's the epidermis, right? And the epidermis is stratified squamous epithelium, right? Once we get below the epidermis, what do we call this? We call this the dermis, right? The dermis is subdivided into two parts. So we have right here below the epidermis is the papillary layer. So it's basically the wavy portion, wavy. And then under the papillary layer, it's the reticular layer. So the dermis is subdivided into the papillary and the reticular. And the reticular is made of one of the tissues we'll see next, which is dense irregular. So all of this tissue you see down here is dense irregular tissue here. And then the papillary portion has our Meissner's corpuscle. Any questions? Is that kind of making sense? That would be a helpful probably picture for you to have somewhere showing you guys the different layers. All right, so going back, looking real quick at our Meissner's corpuscle, you guys now know the location is in the papillary layer of the dermis because it's poking up. So in this picture, what do you see right here? You see epidermis, and then you see epidermis. If we wanted, we could label all the layers again, right? We could say, I don't know, it'd be really nice if I could figure out how to have you guys do it, but boys, right? This would be say, this would be G, girls, granulosum, right? And then that's corneum. Again, boys or basala. Stratum spinosum, boys say, and then granulosum, boys say, girls. Does that make sense? So we're looking at our epidermis. The structure is Meissner's corpuscle, and its function is to detect light touch. So those are how you'd be tested, and then location. Good? Okay. So that was all of our epithelial tissues. So those were all of our epithelial tissues that you see right there. So now we're moving into our connective tissues. So for our connective tissues, this first one is our loose areolar connective tissue. So this is very distinctive. Most students don't struggle at all on the test with loose areolar connective tissue. Again, if you've been studying, if I see a student miss this one, I'm pretty sure that they probably never even looked at it under the microscope. It's very distinctive. So loose areolar connective tissue kind of looks like, to me, it reminds me of The Matrix, like the movie The Matrix. It looks like laser beams, like all over the slide, like all these weird little lines and laser beams. It has these different proteins in it, collagen fibers, elastic fibers, and reticular fibers. You don't need to identify the difference between each of the 3 types of fibers. So I don't know how to, I guess I can like write that in. So don't need to ID the difference between collagen fibers, between elastic fibers, and reticular fibers. If we asked you though, on the lab exam, what are the 3 proteins in this tissue, you do need to be able to say, well, I know that they're collagen, elastic, and reticular. Does that make sense? So we might not say which one is it pointing to right now, but we might ask you the theory question of like, what are the 3 proteins found in this tissue? Good. Location.
another-
Yeah.
Sorry, I have another question. Would you say that one of the like key distinguishing feature between this and the nervous tissue would be that like this has like more fibers? Because
Yeah.
I think I would confuse this with like nervous because I'd confuse it as like seeing dendrites or something.
No, that you bring up a good point. That is like, I would say if you've looked at them maybe once or twice each, they could be confusing. So I'll just, everybody, this is like a great question that she has. So this is nervous tissue. When I show you nervous tissue like this, and then I go back to loose areolar, sometimes this is the best way to study these, is to kind of contrast the ones you're confused on. Do you see how much more is going on here? Like there's lines everywhere, and then the cells are really tiny, like just little dots. And then there's all these lines crisscrossing. It's kind of like chaos. Versus if we go over to the nervous tissue is very like, just here's the neuron, here's the little glial cells. If I pull up another example of nervous tissue, this is another example of nervous tissue that looks probably a little more like our loose areolar, but the thing about it that's distinctive with nervous tissue is it's not consistent. So for loose areolar, it was like it was chaos, but it was all the same across the slide, if that makes sense. Like all over the whole slide, it looks the same pretty much. Versus if you look at your neuron is looking like, okay, we've got this big blob at the center, and then now that it kind of gets less chaotic as we move out from there. Does that make sense? Looking at the 2 next to each other? Great question. I would definitely encourage you guys that if you're feeling confused and you're feeling like, okay, but I keep confusing this one with that one, that actually tells me that you've been studying and you're learning them. When I was taking the class, I was very confused, but I was confused when I started to go over them. If you're not feeling confused, you probably haven't even really looked at anything yet, because they do get very confusing as you start to look at them. So make sure you dedicate that time this week going into the skills lab, getting extra time to look at them. But your starting point should be this PowerPoint and the way I'm going over it now. That'll help you a lot. So loose adipose connective tissue, this is fat. The cell is called an adipocyte. The protein is collagen. The fat vacuole are kind of like these open cells. This is very distinctive. It looks like soap bubbles. I would just say that there is no other slide that looks like this, because they're like these perfect little tiny bubbles. Sometimes I've had a few people say, how do you tell it apart from lung tissue? Lung tissue never has bubbles this small. Do you guys see how big lung tissue is? It's like these large, and they're very irregular in shape. They're not like a circle. They're like this kind of weird shape. Versus if we look again at our loose adipose, they are very circular shape. They look like you sudsed up in the bath and now you have all these bubbles sitting in a pile. Does that make sense? So loose adipose or fat to me looks just like bubbles. It's a perfect way to remember it. loose reticular connective tissue. So this is, to me, I had somebody describe this as cherry blossoms when I was in the class, and it stuck. So I do think that this looks like cherry blossoms. If you picture like on a tree, how they kind of fall on the ground. So these do kind of have that appearance. These are two different slides to show you that sometimes they can be brown or they can be this other stained color. The biggest characteristic here is we just see dots all over the whole slide, because these are found in the lymph nodes, bone marrow, and liver. And that's where basically white blood cells and red white blood cells are mostly stored. So there's a lot of cells all over the slide. That's why the cell is the fibroblast and the leukocyte, fibroblast and leukocyte. So if we look at an example a little closer, this is another example of our loose reticular. And you guys, whoa, do we see how it's little cells all over the whole slide? Yes? These, usually someone will ask me, what are these little blue structures? So those are the actual reticular fibers. So these are our reticular protein fibers, reticular protein fibers. But loose reticular is, again, very distinctive because it is just cells everywhere is how I would describe it. And again, where's our location? In our spleen, lymph nodes, bone marrow, and liver. Any questions so far? That's a good question. Will you be marked down if you put one location versus all locations? You do need to know all locations that we give you to know. So please make sure you hear me. Don't go to the textbook and find all the different locations, because there's like so many more than what we give you to know. But if we give you the location to know, you do need to know all of those, because this, remember, is most likely to be a multiple choice exam with none of these as one of the choices. So for example, the way that I would write this question if I was doing the test is I would say, where is this tissue found? And my options would be spleen, lymph nodes, bone marrow, liver, and none of these. So how would you guys answer that? If this was your tissue that you were looking at under the microscope, and this is the question, how would you guys answer that? What would your answer be? So the lab exam will be some fill in the blank and then some multiple choice, depending on your professor. I know Knudsen is doing mostly multiple choice with maybe possible fill-ins. And then I'm not sure exactly Bender's plan for this semester. So yeah, so E would actually be the correct choice. So somebody put E. E would be the correct choice because the correct answer is actually all, right? All of these are correct. But did we give you an option here that says all? No, right? There's no option that says all of these choices. You cannot choose any of them because all of them are right. So the only option you can choose is none of these, because basically what you're saying is, I'm trying to choose all and I don't have an option to choose all, so I have to choose none. It's not a lot of times we do the question that way, but there is sometimes, I'm not going to lie to you, that the lecture exam and the lab exam are set up. You might have some questions that are that way. If all are true, you choose none. So when you ask, like, do you need to know all the locations that we give you? Yes, because if we give you all those locations and they're all correct, you would need to know to choose all of them. Does that make sense? Most likely that's not the way it's going to be, but that could be a way that we do it, is if it's supposed to be all, you choose none of these choices as your answer. Okay, so that's kind of where the multiple choice can get a bit tricky. So again, loose reticular connective tissue, we have dots all over the slide. Dense irregular connective tissue, this is found in the reticular layer of the dermis, like I had shown you basically up here, right? This reticular layer of the dermis is found down here. So this would be our dense irregular connective tissue. Dense irregular connective tissue. Okay, so that is zoomed in. It looks like this. Okay, so once we zoom in, I'm going to show you this picture. So this is our zoomed in dense irregular connective tissue. In my opinion, it looks like pink ribbons kind of crisscrossing back and forth across the slide. That's how I interpret it. That's what I think it looks like. Maybe to you it looks like something different, but to me it looks like pink ribbons. Try to pull up another example. If we pull up, let's see. If you look at it here and we zoom in on it, do you see how up here, whoa, that's like way too close. This is the epidermis, right? This is the epidermis. This would be dermis. And all of this is dense irregular connective tissue, right? This is epidermis. This is all dermis. And all of this is dense irregular connective tissue. Like I said, it kind of looks like little ribbons all crisscrossing over each other. In case you're wondering, this is a sebaceous gland. Okay. So going back, so
Chris?
that was, yeah,
Can you identify the papillary on that, please?
sure. You mean the papillary layer, right?
Yeah.
So the papillary layer is basically right underneath the epidermis. So the epidermis is the pink squiggly line, right? So this is epidermis. So right underneath that, do you see how it kind of looks like it's fuzzy? Like this little fuzzy part? That would be the papillary layer. As soon as we get below the fuzzy part, we're now in the reticular. All of this would be reticular below that fuzzy part. Does that make sense? Can you see that? Visualize that the fuzzy kind of pink area is the papillary, and then reticular would be below that. You're unlikely, just to be clear, you're unlikely to be asked papillary layer on the exam. I was showing you that because you do need to know what tissues are found in the papillary layer, and you need to identify papillary and reticular on the model. So it was important that you kind of understood the concept. So for papillary layer, what are our tissues? Loose areolar connective tissue. And then our Meissner's corpuscle is located there. And then for reticular layer, it's the dense irregular. So that's kind of the main reason I was showing you those, is I wanted to make sure you guys understood the difference of the locations. Okay. You guys hanging in there okay? Any other questions? I'm gonna keep moving forward. So this is a summary of the skin, kind of the stuff I've already talked about, so you guys could read through that if you want. Basically just showing you the stuff I had kind of drawn earlier. Dense elastic connective tissue. So this is a confusing one. Dense elastic connective tissue is confusing. What you want to look for is the large black wavy line or lines. This is found in the aorta and the vena cava. They are the major artery and vein of your heart. So large black wavy line or lines. So here we see it again, black wavy lines. Again, black wavy line on both of these. Do you see how on the left-hand picture, there's like 50 black lines over here? Versus on this picture, we just see basically two, right? One here and one here. There's kind of a few down here. So on the exam, we could give you either of these pictures. What you're looking for, right? What you're looking for here is the black wavy line, not necessarily how many of them there are, okay? Again, if we go to this next picture here, multiple black wavy lines are all over the slide. So let me pull up a few more examples over on this packet. So you guys have access to all of these pictures, just to be clear, this is all available to you. This is another black wavy line, right? Just one black wavy line, but it's still very distinctive. None of the other tissues have this same black wavy line. Again, black wavy line, and then multiple other little kind of little ones, but there's one major one. Why? Because this is the aorta or the vena cava. So this is what fills with blood when your heart pumps blood out to your body. So if you picture that, it would need to accommodate a large volume of blood. Okay. So that was dense, elastic, connective tissue. Now we have dense, regular connective tissue. This is one of the hardest tissues to identify, and it actually gets most often confused with two other tissues that look very similar. So fibrocartilage and smooth muscle are the other ones that it gets confused with. So we have dense, regular, and then we have fibrocartilage and smooth muscle. So I'll do a compare and contrast. So if we start with dense regular, the biggest thing with dense regular is I made this checklist. So I'll just try to say this very quickly. I wasn't cramming. I had been studying weeks before the lab exam when I took this class, but I couldn't get these three. Like no matter what I did, I just couldn't get it. Dense regular, fibrocartilage, and smooth muscle. It was like, it was going to be the death of me. And I just kept working on it and nothing made sense. And I had like a revelation the night before the exam, and I pulled out pictures and I showed why they were different. So I made a checklist and I'm walking you through that on this PowerPoint so you guys have access to it. So dense regular. So dense regular, you need to know the name, you need to know the location, you need to know the cell and the protein. I feel like hopefully I'm making that clear when I don't necessarily go over it each time. You guys need to study that on your own time. I'm trying to help you tell the difference. So what you're going to see is that this is slightly wavy and it has these gaps in it. Is everybody seeing that? These little like tears, kind of like the one person had said, they look like birds, right? So these look like tears in the slide. So that's where I have slightly wavy with gaps. So this time, these gaps are important and they're something you want to look at. So we see these gaps for dense regular. We see that the nuclei are one color throughout. So if I kind of draw a circle, do you guys see how there's not necessarily like a darker portion? They're kind of just a monochrome, one kind of color throughout. And then you also see that the nuclei are located inside of the gaps. So nuclei are located inside of these gaps. So we'll do another example and show you how that is true again. So this is also dense regular connective tissue. You guys see that there are these gaps all over the slide. Yes, gaps everywhere. The nuclei are one color throughout, and then nuclei are located in the gaps. Okay, so the same thing holds true. And now let's show you a fibro slide. So fibrocartilage is what I used to get the most confused. So initially it looks the same, but what we're going to do is we're going to show you why it's different. So do you guys see the gaps? But do you see how the gaps aren't consistent? So there's like random weird ones up at the top and down at the bottom, but they're not consistent. This is like a massive gap. Yes, like they're weird. And then you might be saying, well, what about this little tiny one? Well, this is where it's very distinctive for fibro. So the nuclei are not found in the gaps if we're looking at fibrocartilage. Fibrocartilage, the nuclei are not found in the gaps. So if you look, I have all these little arrows pointing on the picture, and those are pointing at nuclei of a cell. Do you guys see? And do you see how it's not inside of a gap? It's like basically floating out by itself. Okay, so let's go back and look at a dense regular again so that we can see the difference. So here's our dense regular. Our dense regular has gaps. The nuclei are in the gaps. Our fibro has gaps, but the nuclei are not in the gaps. The other thing that's distinctive about fibro is the nuclei have a dark spot in the lighter area. Dark spot in the lighter area, okay? And it doesn't have the waviness that dense regular has. Here's another example of fibrocartilage. You guys can see that there are gaps, but the nuclei are not in the gaps for fibrocartilage. Versus our dense regular, we have gaps and the nuclei are in the gaps. Fibrocartilage, we have gaps, the nuclei are not in the gaps. So when you guys get to your exam, you're gonna have to pause and you're gonna have to remember this checklist in your head. Okay, do I see gaps? Are the nuclei in the gaps? Yes, dense regular. Are the nuclei outside of the gaps? No, yes, then it would be fibro. This is another example of fibrocartilage. It looks a little bit different here. You can see that it has these gaps and the nuclei are not in the gaps again, okay? Smooth muscle is the third one that this gets confused with. And smooth muscle, this is a good example of one that people get confused. Smooth muscle has no gaps at all. So basically, the biggest way to immediately tell the difference is if you see gaps, you know that it's dense regular or fibrocartilage. If you do not see any gaps at all, that would be smooth muscle. And you can see how the nuclei are kind of in this unique shape and they're flowing in one direction. So again, smooth muscle has no gaps and the nuclei are in this fusiform shape flowing in one direction, okay? And we're just talking about this center portion. Another example of smooth muscle, we do not see any gaps again. Everything's kind of flowing in one direction and these nuclei are long and thin. Another example of smooth muscle, no gaps at all flowing in one direction. And another example of smooth muscle, no gaps at all, and the nuclei are all flowing in one direction. Again, looking at one or two more examples of the fibro. Here's a fibrocartilage example. You can kind of see how there are gaps. This is a very unique fibro example because we can really see the lacuna and the chondrocyte. So I'll show you what I mean. So in this fibro, we can see that there are gaps and the nuclei are not in the gaps for fibrocartilage. For dense regular, there are gaps and the nuclei are in the gaps, okay? Again, dense regular, we see gaps and we see that the nuclei are found in the gaps. For this fibrocartilage, you guys can see that there are these, basically this dark cell, it looks like this, and then it's kind of in this lighter pocket. So this is our chondrocyte in our lacuna. So this is really important for you guys to know. So this is chondrocyte is the cell, and then lacuna is kind of the house where the cell lives. Lacuna is this house. So for fibrocartilage, the chondrocyte in the lacuna for these other pictures that look a little different is the fact that we have this dark spot in a lighter area. The dark spot is actually our chondrocyte and the lighter area is our lacuna. Okay, so dark spot, chondrocyte, lacuna is the lighter area around it. All right, so I'm sure your brain is full, but we still have a few more tissues to get through. I'm not sure.
Really quick.
Yeah, no.
So for the fibrocartilage, what would be like the matrix of that? Because I'm seeing like in the lab packet, like to label the collagen fibers making up the matrix. Does that mean?
The matrix, I believe, is just generalized collagen everywhere. So like essentially everything you see that's not a cell is collagen.
Okay.
So you're not going to have to label that on the lab exam. That's not what we're really interested in you labeling. Is we're looking for, do you know that it's fibrocartilage, which honestly for most people is 90% of the battle, because like I said, it's kind of hard to tell apart. And then location. So the location of fibrocartilage, it has the meniscus of the knee, intervertebral disc, pubic symphysis. The protein, we might ask you what is the protein, but we won't point to it and say, what are we pointing to, if that makes sense.
Okay.
I have a question too.
Yeah.
Just to clarify, like, would we need to know the function of the lacuna and the chondrocyte, like, overall? Or is that not, like, really what we're getting tested on?
That's not really what you're getting tested on. I mean, it's pretty simple. The chondrocyte is just the cartilage cell, and the lacuna is the house. So it is possible, especially, and I'm about to do these, but for hyaline and elastic, we might ask you what is the house for the cell, which is almost like a definition, if that makes sense. So if we say, what is the house for the cell in this tissue, then you'd need to know lacuna. So it's kind of, I guess I'm saying yes and no. Yes, you need to know the function, because you would need, if I said, what is the cell, what is the house for the cell found in this tissue, you should be able to say lacuna. Or what is the cell found in this tissue, you should be able to say chondrocyte. Does that make sense? So
Yeah,
I guess.
I just wanted to know if it was like as specific as we needed to know exactly like what each thing was doing. So yeah, it does make sense. Thank you.
Kind of, sort of. It's like general. I guess it's like very simple. I feel like for the functions, some people feel very overwhelmed by the functions, and I'm trying to be like, I'm being very honest in the functions I'm telling you guys. Like, it's as simple as to increase surface area. That's it, you know, like, but sometimes people will just be overwhelmed by the functions and not study them at all. Whereas I'm literally giving you exactly what is like on the test. Like, that's the exact function, to increase surface area, no more, no less, not more complicated than that. So for hyaline cartilage and elastic cartilage, so these are the other two types. So to tell the difference between hyaline and elastic cartilage, what you're going to do is look at the background. So hyaline and elastic cartilage basically are very distinct. You guys are probably already seeing it. You see how we see these like kind of weird looking white pairs of cells? So these are our chondrocyte in the lacuna, the house. So the lacuna is the house for the chondrocyte, which is the cartilage cell. In hyaline cartilage, there is almost no protein. It is almost all ground substance. So we don't have any protein in the background. It is location you need to know: trachea, fetal skeleton, ends of bones, which is the articular cartilage. Two more examples of hyaline cartilage you guys can see pretty clearly in this picture are lacuna, right, which is our little white circle, and then our chondrocyte. So the chondrocyte is that cartilage cell, and then it lives in a house, and the house is called the lacuna. Okay, so it's that white circle is the lacuna. And as you can see, if you guys all right now, look at the background. Do you guys see how there's no protein fibers really? It just looks like washed out nothing, like a watercolor painting, right? It looks like nothing is really there. Now, when we go over to elastic cartilage, look at the background. Do you guys see how there are now these little black fibers in the background? Those black fibers represent our elastic fibers. So elastic cartilage is found in the ears and epiglottis. Both of those start with E, right? Elastic, ears, epiglottis. So that's kind of an easy memory tool there. But basically with elastic cartilage, how do you tell the difference? You're going to look at the background, and you're going to look for the elastic black fibers in the background, versus in hyaline, it's just a washed out kind of ground substance ground substance only appearance, right? So let me zoom in real quick just to show you another example. So if we look, we're seeing not, well, too close. We're not really seeing any black fibers. I picture it like black hairy moss. We're not really seeing that. This is hyaline. Hyaline, no black fibers. Now, if we look at our other slide, so this is our elastic cartilage. When we zoom in on it, we see these black fibers. Yes, see these black fibers in between the cells. Again, another example, we're zooming in and we see those black fibers. They look kind of like hairs or moss little lines. So hyaline is going to have no hairs. Elastic is going to have those hairs. Hyaline, no hairs. Good? Questions? So that brings us through dense, regular, hyaline, fibro. We kind of talked, we talked about our smooth muscles. So what we just have left is our different muscles and then a few little touch-up tissues, okay? So we're kind of almost done. So I should have a little bit of time to start meiosis. I may have been optimistic about going all the way through both spermatogenesis and oogenesis, but Thursday we'll be able to pick up with whatever we don't finish. And I'm hoping to do the integumentary lecture on Thursday. Or maybe we'll take a vote and do models instead, or part models and part integument. Anyway, basically Thursday, we'll see how it goes, whatever would work best for you guys. I'm thinking maybe I will do lab models first and then do the integument lecture, if that sounds good to everyone. Or we could do some review questions. Basically, if you come, we will do a vote and everyone will get to vote. Very democratic. So for skeletal muscle tissue, well, you don't need to leave. We still have another 30 minutes. I'm still trying to get through stuff. Some people were leaving. Questions?
I do have a quick question. So these sessions are available like for us to watch back, right? Because I'm not able to come in, do the Thursday one.
I don't have a way to record this semester, but I do have some students that are helping me out and they're recording. And I'm sending the sessions and I'm trying to post them in the module for you. So basically just write me on Canvas if you don't see it. I'm hoping to get a recording out to everybody is the shortest way to answer that. I'm not recording because I'm not able to this semester, but other students are kind of pitching in and helping. So hopefully. I can't guarantee it, but hopefully.
Okay. Thank you.
Yeah. Sorry, my recording stuff isn't working and I've got a lot going on and I wasn't able to get it worked out, but fortunately other people are pitching in, so it's working out, hopefully. So for skeletal muscle, this one looks very distinctive because it actually looks like columns or lines going up and down on the slide. So you see that repeated every time you see a skeletal muscle example. One more. So for this skeletal muscle example, do you guys see how we have basically these straight columns up and down? I know they look a little crisscrossed, but mostly it's just up and down, right? And then you can really easily see the striations. So that's one of the things that's very distinctive about skeletal muscle is we see our striations, which is basically made up of our dark A band, which is myosin, and then our light I band, which is actin. So our striations are visible for skeletal muscle, unlike basically cardiac and smooth muscle. We don't really see striations with cardiac and smooth muscle, but with skeletal muscle, they're very easy to see. You can also see that they have, skeletal muscle has multiple we call it multinucleated. They are little tiny nuclei on the outside of the cylinder of the skeletal muscle. So that is very distinctive as well about skeletal muscle. Okay, so going back, looking at it one more time, here's our skeletal muscle tissue example. And we can see that it has the striations pretty visible, even from far away, right here. There are these little lines, and we see the multiple nucleuses on the outside, multinucleated. Okay, so skeletal muscle, pretty easy to tell from other muscles. Yes, I like the raised hand. Amy.
For the striations, there's a specific question in the lab packet that's asking where the striations come from. I wasn't able to find it like on my own. Could you explain
Where
that?
they come from?
Yes.
I think it's just what I think it's basically what I just said. Dark
Okay.
A band comes from myosin protein, and then you could say light I band comes from actin protein. And
Okay.
then asking where the striations come from, the striations are light and dark. So it would basically be like, what I'm trying to say is a dark line, a dark line, a dark line. And then if you guys look, that's what this is. See how there's a dark and then a light and then a dark and then a light?
Mhm.
The dark part is myosin and then the light part is actin.
Got it. Okay. Thank you.
Okay, so we'll do cardiac muscle. So cardiac muscle is typically a harder one, but I'll show you kind of how to tell it specifically. So cardiac muscle basically has this branching appearance. So when we look at more examples, you'll see it. It looks like it's branching everywhere, kind of all at once. Here, it's very straight, but it still has that branching appearance. The other thing that's very distinctive about it is two more things. It has nuclei, I have it here, that look like, to me, like cans of soup. So they aren't stretched out. They're kind of short and fat. So if I draw a circle, you see what I mean. Do you see how it's kind of these short and fat nuclei all over, right? Versus like the skeletal muscle, they were really tiny and almost really hard to see. And then in smooth muscle, which is something that cardiac gets confused for, the nuclei are very long and thin, and we call that fusiform, okay? So for smooth muscle, long and thin, fusiform is the name of that, okay? And they're these long and thin nuclei. Whereas in cardiac muscle, the shape of the nuclei is much shorter and fatter. The other thing that's distinctive about cardiac muscle, and we will give it to you if you have it on the exam, you will see it, they are called intercalated discs. So if you're on your exam and you see an intercalated disc, so this is one right here. This is one, they are like a dark line pretty much that divides one cardiac muscle tissue cell from another. And an intercalated disc is distinctive to cardiac muscle. Their function is to allow sodium to rush into the cardiac muscle cell. Okay, so that's the intercalated disc. We will, if you have cardiac muscle on your exam, it is very likely we'll have this black pointer, right, pointing to an intercalated disc. It is not pointing to one in this example, but it would be on your exam, right? What is the structure at the end of the pointer? An intercalated disc. What is the tissue? Cardiac muscle, right, located in the heart. So to show you on an additional slide, if we looked on this one, we see a lot of intercalated discs, right? Here's an intercalated disc, here's an intercalated disc, here, one more down here, another one. So all of those are intercalated discs. Again, this is our cardiac muscle. Cardiac muscle has our intercalated pillated disc. That is the biggest distinguishing feature. In addition to, it kind of looks like branches, and it also has larger nuclei than the smooth muscle or the skeletal muscle. Okay. I'm kind of thinking about just doing some models since we only have 15 minutes. Thinking about doing some models and then on Thursday focusing on the meiosis and then maybe some review questions for the lecture exam. That sounds good to everybody. So I'm thinking I'm going to do some of the lab models right now since we're kind of in the lab exam mode. I feel like that might be a good use of our time. That sounds okay to everybody. So that was the end. Oh no, never mind. I still have these. So bone is another example of a tissue. You guys need to go over these functions, but I will label them for you just because you do need to be, you are responsible for these on the lab exam. So I want to make sure you guys have them labeled correctly. So for lamellae are the rings, like the rings of a tree trunk. These are the round rings that go around bone. So these would be lamellae, okay? So I'm going to kind of draw a little arrow, lamellae. A lacuna is the house for the osteocyte. These little black structures, those represent your lacuna. Osteocytes live inside of the lacuna, so we can't really see them. So that's why I'm going to just draw an arrow basically for the same area. So the way we'd ask you that is, what is the structure? And you would say lacuna, and then maybe you would say what lives inside the structure, and you would say osteocyte. Does that make sense? If it's pointing to the black circle, you would, the black little structure, you would say lacuna, and then what lives inside is the osteocyte. Canaliculi are the cracks in the bone that basically join or connect the different osteocytes living in their lacuna. So these little cracks right here are the canaliculi. So those little cracks, they kind of look like hairs. Those are the canaliculi. The Haversian canal is the central circle. So this is the Haversian canal right here. It has the blood vessels and the nerve. And then the hydroxyapatite is the hard ground substance that makes bone hard. So it would just be kind of like everything, a generalized background, everything. Bone, it's very, I'll just tell you right away, it's very unlikely we will ask you, very unlikely you will be asked, what is this tissue? This is very distinctive. Have I shown you a single other tissue that looks yellowish orange like this? They all look exactly like this. So we're not going to really ask you, is this bone? That's easy. We're going to be asking you guys for some of these terms and their functions. And just one last thing on the bone, osteon or Haversian system is the name of the entire thing that I just finished labeling. So everything that we labeled, each one of those is an osteon. So you can see there's another osteon over here. There's another one over here. There's another one right here. Each one, each like big unit is an osteon. Does that make sense? So that's bone. So that would be wrapping up bone. And now for blood. So for blood, we have our white blood cells, our red blood cells, and our platelets, right? White blood cells, red blood cells, and platelets. Red blood cells are going to be all of these pink, right? They're also called erythrocyte. The white blood cells are the purple little guys, and those are also called your leukocyte. You have 5 different white blood cells, and you guys do need to know their functions for this lab exam. So we memorize this using the mnemonic, never let monkeys eat bananas. Never let monkeys eat bananas. So the 5 types of white blood cells, neutrophil for never, lymphocyte for let, monocyte for monkeys, eosinophil for eat, and then basophil for bananas. Never let monkeys eat bananas. So this mnemonic orders the leukocytes in commonality from most common to least common. So never is the most common leukocyte. Neutrophils is the most common. Basophils are the least common. We may ask you on the lab exam, what is the most common white blood cell? You would say neutrophil because it's never let monkeys eat bananas. What is the second least common white blood cell? And you would answer eosinophil because never let monkeys eat bananas is the second least common. Okay. You also need to know whether they are granular or agranular, granular and in fill. So neutrophil, eosinophil, and basophil are all granular, agranular and in site. So lymphocyte and monocyte, both of those are agranular. For functions, you can use this mnemonic right here. And I wrote them all out for you. So you use the BAMF mnemonic. So for never let monkeys eat bananas, we can use BAMF and you can draw it out next to it. And I tried to write all of this here for you guys to have it as a resource. But basically for neutrophils, they would phagocytize bacteria. That's the B. Lymphocytes have the A antigen antibody complex. Monocytes become macrophage, M. Eosinophil phagocytize parasites, P. And basophil secrete histamine heparin, B. It goes with H. Someone raised their hand. Sorry, I was just finishing my thought there. What's up? Did you have a question? Sorry, I'm not sure who raised their hand. Okay. Any questions at all?
Sorry.
Yeah.
So the mnemonic is in order from most to least common?
Mhm.
Is the mnemonic in order from most to least common for the white blood cells?
Yes. So the neutrophil would be the most common, and then the basophil would be the least common. All right. Any other questions on this? Before we go, I'm going to show you guys those on the models because I think that would be a good thing to wrap up with, the models of the white blood cells, because we'll for sure be testing you guys using those white blood cell models. We have those on every lab exam. We put those on there. Okay, so the nervous tissue is our last tissue. So this is located in the brain, spinal cord, and nerves. It has these neurons right in here with our neuroglial cells. So the neuroglial cells are these little cells that kind of surround the neuron here. They are these little like blue dots. And then the neuron is this main portion here. It has the dendrites. So these are our dendrites. So I'll write a D. And then it has our axon coming out. And then of course it has the cell body in the middle with the axon hillock. And this would be our axon hillock right here. Okay, so that's kind of some stuff that you were asked to draw and label in your packet. So you guys are welcome to use this picture and work on drawing and labeling that in your packet. Remember, use your time in lab wisely. Do not waste your time in lab drawing pictures and answering questions. Spend your time at home drawing pictures and answering questions, and go into lab trying to practice distinguishing these tissues. The best idea that I might have for you, if you have a lab partner in lab, get that lab partner and have them quiz you. Say, hey, will you pull up? I'm going to work on pulling up the models while I say this, but say, hey, pull up a tissue and quiz me on what it is. Don't tell me what it is and quiz me. And then you do the same thing for them. Pull out a slide, focus it, and show it to them, and quiz each other on what each one was, what each one is. Does that make sense? So study them at home, study them tonight, go into lab tomorrow and quiz a friend. Find a friend, quiz a friend. Other questions while I pull up these models, because we'll finish with models. The tissue slides are in the study resources module under lab exam number 1, practice exam PowerPoint. Lab exam number 1, practice exam PowerPoint. So remember, so far in SI, I've gone over the mitosis models. I've gone over the cell model. I went over mitochondria and phospholipid bilayer. So now we're going to work on the tissue models, the intestine models, and then I'm not sure exactly all that I'll get through, but I'll go through as much as I can before we end. And then whatever models I don't get to, I will go over on Thursday, and then we'll also go over some meiosis and integument, okay? So don't miss Thursday's SI. Meiosis is a very challenging topic for students on the lecture exam. Okay, so this is going to be our smooth muscle model. So smooth muscle model. And what do you need to know on it? You need to know, first off, that it's smooth muscle. And then you need to know that the shape of the cell is fusiform. So what is the shape of the smooth muscle cell? Fusiform. This means small, large, small, or like squished. So smooth muscle, shape of the cell is fusiform. Good. That's all you need to know for this model. Very simple and straightforward. All right. For this model, you again just need to know what tissue it is and then where it's found. So this is simple cuboidal epithelium. It's going to be located in the kidney. Good. This model is our skeletal muscle. So obviously you need to know that it's skeletal muscle. And then you would want to know the striations. So what I had already talked about, now we're going to ask you guys to label them to make sure you understand the concept. For the exam, you could be asked to label the striations. So remember, dark / A band equals what? Myosin, right? Whereas light / I band equals actin. Light / I band equals actin. Okay, so you might be asked to label those on the exam. So skeletal muscle, dark A band is the dark lines, light I band or actin is the lighter portions in between the dark lines. Any questions at all?
Is that all we have to know for the model for skeletal muscle?
Yes, that's all you need to know.
Okay, thank you.
Yeah. We have another skeletal muscle model. So for this skeletal muscle model, same things that you need to know. It's just they look a little different now. So what you're going to see are the these darker, the 2 red lines is going to be your dark band, A band equals myosin. That will be the 2 red lines. The white or lighter area, that's going to be your actin, I band, your light, I band, actin. Again, overall, this is still the same thing again. This is our skeletal muscle, muscle model. Good. All right. So we also have a cardiac muscle model. So what do we need to know on the cardiac muscle model? Striations again. So you could be asked, are striations again? So this is our dark A band, dark A band myosin. And then next to it, these little light portions here, light I band actin.
So, if you don't mind me asking a question. Yeah. Muscle and skeletal tissue both have the same bands, even though it's in different parts of the body.
They are both considered a muscle. So skeletal muscle and then cardiac muscle, because they contract, you'll learn about it, not this unit, but next unit. The way that muscle contracts uses actin and myosin. So even though, yeah, they're different parts. One is cardiac muscle. When it contracts to pump your heart, it's going to use actin and myosin, just like your muscles that move your legs. They do it a little differently, but they basically use the same process.
Okay.
Good question. Yeah. Yeah, if anybody has questions, feel free to interrupt or ask at any time. I appreciate that. The questions are what helps us all learn, for sure. So the arrow that I just drew is intercalated discs. These move sodium through the heart muscle rapidly. You definitely, if you get the cardiac muscle on the exam, you will be asked about the intercalated discs. If you get cardiac muscle on a slide on the microscope, you'll be asked about intercalated discs. So make sure you understand what those look like under the microscope and on the model, and then kind of how to spell them, what they are. Okay. So that was the different tissues. So the different muscle types and how to identify and label those. So let's do the intestine, and then we'll probably go like 1 or 2 minutes over 6:45, and then I'll let you go. Okay, but I know we started a few minutes after 4:45. So this is our intestine model. So what we want to look at is what we're expecting you guys to label on the test. So it's all the same stuff that we learned about with our simple columnar. Remember, what is the tissue that actually lines this model? What is the tissue that lines this model? We know that this is a model of the intestine, and we know that that means that it's simple columnar epithelium, right? So that's our tissue that we see here. Remember our different structures we already labeled on the microscope. What were our little circular structures called? Goblet cells, right? Goblet cells were our little circles. So goblet cells, once again, we need to label them on this microscope. So they secrete mucin or mucus to keep the intestine moist. You need to know that function for the lab exam on the model and on the microscope for the picture. The blue structure, let's grab a different color. The blue structure you see right here, this little blue structure, is our lacteal, right? Our lacteal absorbs fatty acids or lipids, right? So if you eat food and it gets into your intestines, the fatty acids or lipids from the food is getting absorbed into the lacteal. What about our capillary bed? This over here, this kind of webbing of red vessels is the capillary bed. And remember, we said that this absorbs carbs, nucleic acids, and proteins, again, from the food that you eat. And now, well, I can label this real quick. This is a lymph node. So this would be a lymph node. Okay, so here's a little bit of a critical thinking one. First, I'll label the villus. So remember this entire structure here, we call that entire structure a villus. And what was the function of the villus? To increase surface area. Okay, so what is the name of the hair-like structures that line this tissue. So are they So let's all answer this question. What is the name of the hair-like structures that line this tissue? Okay, good. If you're putting E, what is the correct answer? Somebody chime in. What's our correct answer? Microvilli, right? So microvilli are the hair-like structures that line the intestine. Cilia are the hair-like structures that line the respiratory tract. Great job, guys. Nicely done. Everybody got that. Really good. So microvilli, what do these do? Slow food down for absorption. Right? And so what is the name of the hair-like structures that line this tissue? Microvilli. Okay? All right, so I will see you guys on Thursday. We will finish up our models and we'll do meiosis. Don't miss it. It's highly missed on the lecture exam, and we'll hopefully get to some practice questions on Thursday. Thank you guys so much for coming, and happy studying. Bye guys, thank you.